Dr. Stephen Choi is the recipient of one of Prostate Cancer BC's 2022 research grants and will be speaking on his award-winning research. He is a postdoctoral fellow under the supervision of Dr. Yu-Zhu Wang, a leading expert in the field of prostate cancer modeling. Stephen first began working in Dr. Wang's lab as an undergraduate student in 2010, investigating how cancer cells evade immune detection. He returned to Dr. Wang's lab upon graduation to search for an effective cancer treatment that would address this fundamental characteristic of cancer cells. He began exploring the relationship between altered cancer cell metabolism and anti-cancer immunity, and he eventually wrote his doctoral dissertation on the critical role of MCT4 in suppressing anti-cancer immune response. Since that time, Dr. Choi has focused his research on developing an effective inhibitor of MCT4 for the treatment of advanced cancers. He has co-authored a number of papers on the role of MCT4 in cancer biology. And now I will turn it over to Dr. Choi.
Hello, everyone. Thank you for having me and giving me this opportunity to share my research. And so I will have slides to present. Is that going to be something I can do? All right. So let's see if yes. Can everybody see this as I'm presenting?
That's great.
All right. Sorry, it's been a while since I've used Zoom, so be patient with me. Thank you.
No worries.
All right, so yeah, like I said, thank you so much for having me this morning to present my research. And of course, thank you PCFBC for the support that they have been giving, especially in terms of the award and funding the research. And so there's a lot of work that I'm able to do because of the support that they have shown. And so I'll just, today, knowing it is the holidays, so I'm not going to give like a very, very detailed sort of presentation of my research, but I will just give you a broad overview of it, give you some of the details to the experiments that we've been doing, and to hopefully give you an idea of some of the future directions that we're hoping to take this project as we move forward. And so to begin with, like what Rochelle was saying earlier, I've been working on this project for probably close to 10 years at this point, and it started when I was a graduate student with Dr. Wang. And so at that point in time, being like a young student, he challenged me to take on a project that was a little bit different than a typical research project. And so at that point in time, he was saying that, well, there's a lot of research, clearly important research work in cancer. And a lot of times the analogy can be, it's like trying to trim down a tree. And so you can imagine that here's a tree, and then so there's different kinds of cancer, there's different subtypes of cancer, there's different cancers with different kinds of mutations and unique characteristics. And so there's a lot of research that focuses on one particular subtype, and it's kind of like cutting off a branch. And of course, that's very important. That's very necessary research work to do. But as a young and fresh graduate student, my supervisor was like, well, hey, why don't we try something different? And if it doesn't work, we can go back to something that's more conventional. But he was saying that, well, maybe there's something that's fundamental to multiple kinds of cancer. Maybe there's some unique characteristics that is, yes, cancer specific, but is kind of broadly applicable to many different kinds of cancers, including advanced prostate cancer. And so again, using this analogy, at that point in time, the research was framed as, well, instead of cutting off a branch up at the very top, whether we can find a therapeutic strategy or find some cancer characteristic that hits cancer at where the trunk is, at some place where that's more fundamental to cancer survival, more fundamental to being able to stop cancers from growing. And so the overall idea of this project is that, well, MCT4 lactic acid is something that falls closer to the trunk of this tree, something that's important and fundamental across multiple kinds of cancer. And so the hope is that with effective therapeutic strategy here, we would be able to sort of address and to inhibit cancer by these many different things that we know as cancer hallmarks. And so what these hallmarks are is that these are cancer characteristics that make cancer cells different than normal cells. And so, for example, we know that normal cells will need to proliferate at some point in time to, say, for example, replace lost skin and things like that. But cancer cells sort of replicate and grow in ways that are less controlled and causes problems in the body. And so there's all of these different characteristics when it comes to cancers that we look at. And we're hoping that with MCT4 inhibition, we'll be able to stop. And so the primary ones that we want to focus on is there's going to be the ability to stop cancers from evading an attack by the immune system. We're going to be able to stop cancers from spreading and metastasizing. That will be able to stop cancer's abnormal metabolism. That will be able to sort of reduce the resistance to treatment, and we'll be able to also reduce the growth of blood vessel. And we're hoping to do all of this by targeting one fundamental aspect of cancer biology. And to give you an overview of what that fundamental aspect is, it began with a study on cancer metabolism. And so metabolism is just a fancy word for describing how cells are able to get energy. And so we know that all forms of life will require some energy in some way, shape, or form. And normally in humans, and especially in our cells, the process here is to take glucose and then to break it down into carbon dioxide using oxygen. And so that's what's typically known as normal metabolism. And then this generates a lot of the energy that a normal cell would need. But for maybe close to even 100 years now at this point, is that people have known that cancer cells use energy differently. Cancer cells would take glucose, and then cancer cells would then break it down somewhat, like maybe halfway into a compound known as pyruvate. And then instead of breaking that even further into carbon dioxide, this pyruvate gets strangely converted into lactic acid. And so again, for close to 100 years, people just thought that, well, lactic acid is kind of like a waste product, kind of like the garbage that you would generate on a day-to-day basis. You kind of take it out of your house, and then once a week or maybe once every 2 weeks, the garbage truck comes and picks it up. And people just thought, okay, well, lactic acid was a waste product. There's no point, there's no need to look at it any further. And then for 80, 90 years, people just kind of left it at that. But from my research at that point in time, looking into some of the hints in the literature, it seems like, well, lactic acid isn't quite just a waste product. That it seems like, well, a lactic acid, and especially lactic acid being taken out of the cell and just kind of left floating around in the tumor, this causes what is known as an acidic tumor microenvironment. And what this acid does is that it affects a lot of the downstream processes that I was talking about earlier. Like an acidic tumor microenvironment is able to sort of help the cancer cells make more blood vessels. It's able to help cancer cells metastasize. It's able to help cancer cells stop the immune system from functioning properly. And so at that point in time, it just seems like, well, lactic acid maybe is something that, conventionally speaking, conventional scientists have just sort of like forgotten and ignored. But if we take a deeper look at it, it is actually one of those fundamental characteristics of cancer that we can hope to target, and we can hope to, by targeting it, arrive at an effective therapy. And so another sort of very closely related and important aspect of this, and I've mentioned a couple of times already, is the ability of sort of our own immune system to be able to kill cancer cells. And so again, this is just a generic sort of picture that kind of describes what may or may not happen, but that normal cells somehow through mutations and changes becomes cancer cells. And in a lot of circumstances, actually, our immune system is able to sort of detect these changes, detect these cancer cells, and get rid of them. And then so cancer doesn't develop. But every now and then, there are some cancer cells that can't be eliminated by the immune system. And there's sort of a phase that's called equilibrium. So there's some cancer cells, there's some immune cells that are working. But then over time, what happens is that the cancer escapes immune detection. So the cancer is able to avoid being destroyed, and then it'll just keep growing and growing. And so we found that lactic acid is actually one of the molecules that is very important to all of this process, into this entire process, in that when there's a lot of lactic acid when the outside environment becomes very, very acidic, then the immune cells are not able to work as well. And then this allows the cancers to continue to grow. And so at that point, we published a couple of papers sort of describing our hypothesis. And earlier on in sort of like 2013, 2014, that was still just at the hypothesis stage. And then so once we've generated our hypothesis, then we can begin doing some experiments to see, well, whether what we propose and what we suggest was actually real or maybe it wasn't. And then so of course, as the years went by from 2013, 2014 to now, like 8 years later, there's more and more people, like whether it's our own research that we've done, or whether that's just in the field in cancer research, more and more people have confirmed the fact that an acidic tumor microenvironment, as caused by the sort of excessive generation of lactic acid, is actually something that is very, very important. And so again, focusing on the immune side of it, now we know that, well, there's many different immune cells that are involved in sort of anti-cancer immunity. And there are many different immune cell types that can be affected by lactic acid. And so generally speaking, there's two groups of immune cells. There is one that we call the effector or the cytotoxic immune cells. And these are the ones that actively kill cancer cells. And then there's like the sort of seesaw, the balance of it. The other side of it is what we call the suppressive immune cells. And these are the immune cells that stops the immune system from activating. And you could imagine that in a normal circumstance, that's actually important. So if we get the flu, or if we get a cold, or if we get COVID or something like that, like we do want our immune system to attack the virus, attack the cells that are infected by the virus, and to get rid of the infection. But at some point in time, once the infection clears, we need to be able to stop the immune cells from keep attacking other things. And so that's where the suppressive side comes in, is that generally speaking, in an infection, the cytotoxic, the activated side of it works. And then once the infection clears, then the suppressive side comes in and starts working, and then it stops. And then you kind of get the clearing of the immune response. But what we find in cancer is that, well, there's a very highly active suppressive side of things without a very active cytotoxic or active side of things. And so lactic acid is involved in sort of tipping that balance in favor of a suppressive immune environment. And so this allows cancers to continue to grow, to continue to proliferate, and it stops our normal immune system from being able to kill these cancer cells. So when we sort of take all of that together and combine it into, well, what's the underlying theory that that sort of directs our research today? It is boiled down to the fact that cancer cells make a lot of lactic acid. And so if we're somehow able to, by a drug, by a treatment, inhibit this lactic acid from coming out of the cell, we would have many, many downstream effects. And so some of them, we call them direct effects on cancer cells. And so this will inhibit cancers themselves. So it will cause a backup of lactic acid so that inside the cell, it becomes very acidic, but also it will block the metabolism of cancer cells. But we're also hoping that there would be a second side, an indirect, what we call an indirect or an effect on the tumor microenvironment. And that this would sort of involve stopping new blood vessels from being made, reducing the amount of invasion and metastasis that happens, but most importantly, then restoring this anti-cancer immunity that our bodies like normally have. And so the hope of this project overall is that by these different effects, whether it's direct effect, whether it's indirect effect, it'll all funnel into killing cancer cells more effectively. And so one of the very important targets that we're looking at is a protein that is called MCT4. And so proteins are just part of, like, you can almost think of, if you think of a cell as like a giant robot, for example, or a giant machine, these proteins are different functional parts to it. So maybe here's a screw, and maybe here is maybe a conveyor belt of some sort, and here is like a welding equipment and things like that. And so MCT4 is one of these proteins, and it is one of these proteins that sit on inside the cell membrane. And so what a cell membrane is just kind of think of like an eggshell almost, where sort of outside the eggshell is just outside, and then within it is the cell. And so these are called channel proteins that are in kind of like in the eggshell, in the cell membrane, that gives it a little bit of a hole such that it can have things come in and go out. And so in this case, MCT4 is responsible for taking lactic acid that is inside the cell and then transporting it, moving out, moving lactic acid out into the tumor microenvironment. And so what we know is that more MCT4 expression is generally a bad sign. And we can see this across many, many different kinds of cancer. So prostate cancer for sure, but we also can see this, or other people have studied this and have shown that, well, there's more MCT4 in sort of aggressive breast cancers, in aggressive liver cancers, and so forth and so forth. And so our hope is that by developing an effective MCT4 inhibitor, then we can have, of course, an impact on prostate cancer patient survival, but also then like looking a little bit beyond that to other kinds of aggressive cancers as well. So when it comes to the experimental strategy that we're doing, so everything else before that, that was kind of the project background that we were working with. And so kind of with our hypothesis, with all the background that we have, we will need to kind of narrow down to, well, we will need to find a strategy to actually develop a drug now. And so when it comes to drug development, there's a couple of approaches. The most standard approach is to have maybe a team of 100 chemists or something, and then have a robot that would then help you test billions and billions of compounds to just eventually find the one that works. And this tends to be how big pharmaceutical companies like think Merck, think Pfizer, think AstraZeneca, that is how they develop their drugs. But of course, as a graduate student in an academic lab, we do not have 100 chemists. We do not have billions of dollars of robotic equipment. And so asking me and maybe asking like a handful of team members to let's screen 1 billion compounds in the lab and do experiments of them is going to be very, very time consuming. It's going to be a lot of work. It's pretty much impossible to do. And so in order to change the way of doing things, in order to still be able to develop a drug for this, we looked at a computer. We turned to a computer model. We turned to a computer programming to help us do a lot of this sort of guess and test work. And so what we're able to do is we're able to sort of predict what shape the MCT4 channel looks like. And then from there, predict what kind of molecules are going to be able to fit kind of in the middle of this tunnel. And then you can block it from doing what it's supposed to do. So you can imagine kind of any kind of tunnel, if there's a car crash right in the middle of it, well, the traffic is going to stop flowing. And so that's kind of the same idea. If we're able to find a molecule that blocks this tunnel, then the protein will stop working. And so overall then, in terms of how these experiments work, is that we begin with the modeling. So we begin with finding the shape of what the MCT4 protein looks like, finding sort of in the middle of it, what compounds may or may not fit. And then from there, the computer will spit out a list of compounds that the computer thinks will most likely be a good drug. And then so that way we can go from like 1.6 billion compounds, and we can come down to maybe 100 compounds that we test in the lab. And so then 100 compounds is much more manageable than 1 billion. And so then whether that's myself, whether that's other team members, we would then go and test these 100 compounds to see, okay, which ones work, which ones don't. And then we'll have this process where it's called iterative optimization, where we will say, for example, test 100 compounds and say 10 of them worked. Then we'll take it back into the computer and tell the computer, hey, these 10 compounds work. Can you make better compounds and optimize these compounds for us? And the computer will give us a new list. Oh, maybe there's 50 compounds now. And then we will come back into the lab and do some more testing on them. And so over the years, we're currently on our 20th round of optimization. And we've been testing a good, like almost 800 compounds at this point in time. And we have some very, very promising candidates. We have some compounds that are working the way that we're hoping it would work. And so, of course, for us, we're hoping to be able to take this and then optimize it further to test it further, and then eventually to be able to bring it into clinical trials for patients. And so, like I mentioned, there's a lot of work that is involved. And so it's not just me and not just myself who's been generating this data and making progress, but it's, of course, with a team of other people. And what we do in the lab is that we have a whole panel of tests that we run each compound through. And so there's a whole bunch of criteria that we want to make sure we assess, such that we know that it's an MCT4 inhibitor and not, like, say, rat poison. And then we'll run through these tests, and then we will be able from there to pick the most promising compounds. And so just so not to go into too much details of it, I will just give you a couple of the most important experiments that we run, and then of course tell you that, yeah, like based on the experiments that we've run, we do have some very good chemicals that we think could be very good drugs as we optimize them further and develop them further. And so one of the most important assays that we run is that we want to make sure that our compounds are able to stop lactic acid from leaving the cell. That's going to be the fundamental, the most important part of all of our hypotheses. And so we do this assay called an intracellular lactate assay. And so we measure just how much lactic acid is inside the cell. And so there's a different kind of cells that we use to make sure that the assay is working properly. There's different kind of compounds that we test against to make sure that, well, our compounds are working just as well as the other ones. And so one of the important parts of this is that MCT4 is not the only lactic acid tunnel. There is another major one called MCT1. This one is kind of everywhere, all over the body, in every tissue you can imagine. And so we, of course, don't want to be targeting something that is quite so widespread in normal tissue, because, well, that increases the chance of toxicity. And so we do want to focus just on MCT4, but we have to make sure that our compound doesn't hit MCT1. And so we have these cells that will work only using MCT1, and there's an MCT1 inhibitor that AstraZeneca has developed. And so we're able to see that, okay, well, as we give more and more drugs and over a longer period of time, then the cells that use MCT1, given an MCT1 inhibitor, kind of like has an accumulation of lactic acid inside the cell. Whereas in another cell that has MCT1 and MCT4, we see that, okay, well, maybe there's not as much effect. And then in cells that only have MCT4, then using an MCT1 inhibitor has no effect at all. And so it's kind of along the same lines that we start evaluating our own compound, and that we're able to see that, well, say, for example, in compound 327 and compound 384 and 387, we're getting an inhibition of MCT4 in the red, sort of the MCT4 only cells, but then it's not really affecting the MCT1 cells or the MCT1 and MCT4 cells. And so this is something that gives us confidence that, okay, well, our compounds are working the way that they're supposed to work. And of course, over the years, over the course of the optimization, we're able to actually improve these compounds quite substantially. So you can see here that, well, if 100% is baseline, our starting compounds were able to inhibit lactic acid maybe by 3 times, maybe 6 times. But now we are able to inhibit it by maybe 15, 16 times. And probably the most promising thing is that there are other competitor compounds out there, and our compound is able to do better than the other ones, almost like twice as good. And so this helps us and gives us confidence that the work that we're doing is on the right track, that the assays we're using are good assays, but that also the computer modeling is a very good and effective approach. Another assay that we use is what's called a cytotoxicity assay. And so, of course, ultimately, the end goal is to kill cancer cells. And so we want to make sure that our drug is able to do that. But we also need to make sure that our drug doesn't just randomly kill any sort of cells. And so we then do a series of validations for these compounds to make sure that they're only killing the MCT4 sensitive cells and sort of sparing the other kind of cells. And so generally speaking, there's a couple of categories that we can categorize our compounds based on. So there are the ones that are toxic. And so these are the ones, as you can see, well, it's the color red is bad, right? Green is good, so to speak. And so you can see that it's killing cells across the board, like no matter what cell it is, no matter whether they use MCT1, MCT4, everything is just done. And so in this case, we can be like, well, okay, a compound is probably just a toxic compound and not a very good drug. And alternatively, the other category is that it's just an ineffective compound. And so if you see no effect all across the board, like none of the cells are dying, all the cells are completely happy, then say, okay, well, maybe this is also not a very good drug. This is categorized as a not an ineffective compound. And sometimes what we're able to see is that, well, we have some compounds that inhibit MCT1, but not MCT4. And so we're like, okay, well, that's kind of the opposite of what we're hoping for. And then sometimes we see that they inhibit both MCT1 and MCT4. But ultimately, the compounds that we want to be focusing on are the compounds that inhibit MCT4 only without inhibiting MCT1 and without showing toxicity to just normal generic cells. And then finally, the last most important assay that we run is called a co-culture study. And so what we're able to do in the lab is we're able to, from healthy volunteers, isolate immune cells and then activate these immune cells in the lab so that they would kill cancer cells in the lab. And then so what we're able to do is that if we're able to take pictures and sort of observe these cells over time, we will be able to see that these immune cells then ends up killing the cancer cells. And then we can start adding drugs, we can start adding immune cells, again, just to see how the different components that we're adding into this behave. And so one of the most promising data that we have so far is the fact that our compounds are able to reactivate anti-cancer immunity. So what we're able to see is that, well, if we just leave the cancer cells, leave them alone and let them grow, this would be the blue line where there's just more and more and more and more cells until sort of the entire plate is covered with cancer cells and they stop growing. And so that's the blue line. What we're able to see is that, well, if we added some immune cells in there, which is the black line, we're able to delay the growth by a little bit. And so that the immune cells are able to kill some of the cancer cells, but ultimately they get to the same place. So like there's some cell killing, but the cancer cells are able to overcome the anti-cancer immunity, and then they kind of reach the same place just a little bit later. But what's important to note, and what's important for our research, is that, well, if we add in our drug, in this case, say, for example, compound number 387, we see that, well, there's maybe some inhibition of cancer growth, kind of at a halfway point. And this is what we, on the one side of it, we called initially the direct killing effects, so that our compound is able to directly kill the cancer cells, and there is a reduction in cancer growth. But the most important part of it is when we add the compound and we add the immune cells, then we're able to get a complete killing of the cancer cells that are there in the dish. And so what the show is that the other side of it, the sort of indirect effect is just as important. That by inhibiting lactic acid excretion, by inhibiting lactic acid production in these cancer cells, yes, we're able to have some effects on just the cancer cells themselves, but we're also able to reactivate anti-cancer immunity. And so this is, of course, the data that we're working with, the compounds that we're seeing, and then, of course, we're hoping that, well, all of these together will lead to better and more effective therapeutics as we continue to research and continue to develop and optimize these compounds. And so just as a quick summary of the work that we've been doing, the work that PCFBC has supported throughout these years, our current best compounds are able to inhibit lactic acid discharge through MCT4, and it is also able to inhibit cancer cell proliferation. And so initial testing of these compounds show that it is able to, on the one hand, cause a direct killing effect on cancer cells, but on the other hand, reactivate anti-cancer immunity as an indirect effect. And so our testing in animals and things like that generally suggests that these compounds are safe or non-toxic and only have a cancer-specific effect. And so, of course, our hope is to continue to optimize these compounds, continue to improve their efficacy, the specificity, and of course their stability, and then overall then confirm their safety. We're hoping to use some animal models in order to do that. And then at the end of the day, we do hope to be able to take this drug into clinical trials in advanced prostate cancer patients, and then expand that eventually to patients of other aggressive cancers. And hopefully at the end of all of this research work, we'll be able to have a compound that is effective. That's an effective therapeutic strategy. And so yeah, I just want to take this time to thank all of you for listening, to thank PCFBC and all of the grant funding agencies that have supported our work through the years. Thank the team and thank the lab. And I'll be happy to take any questions.